Excretion refers to the elimination of waste products from the body in order to maintain homeostasis and protect the body from their toxicity. It helps to maintain a constant body temperature by removing excess heat and maintain a steady internal environment in association with the other systems of the body. Parts of the urinary system in mammals. We shall now see the urinary system of human beings as a representative example of the excretory system in mammals. The components of the system are a pair of kidneys, a pair of ureters, a urinary bladder and an urethra. In mammals, the kidney is metanephric type and is also posterior kidney. In adult and mesonephric type in embryonic stages. Kidney is mesodermal in origin and develops from the nephrostomes of early embryo. The kidneys are dark bean-shaped organs about 4 to 5 inches long. They are located on each side of the vertebral column in the abdomen. The kidneys are situated below the diaphragm on the left and right sides and are partially protected by the 11th and 12th pairs of ribs. The right kidney is lower than the left because the liver takes up much space on that side. Each kidney is surrounded by layers of fibrous connective tissue and a mass of adipose tissue which cushions it and holds it in place. On the medial side, each kidney has a concavity called hilus or hilum. The blood and lymph vessels, nerves and ureters enter or leave the kidney through the hilus. The mammalian kidney consists of an outer cortex and an inner medulla. It is composed of functional units called nephrons or uriniferous tubules. We shall see the blood supply to the kidney. In each kidney, the oxygenated blood from the renal artery enters from the dorsal aorta. This artery divides into smaller arteries in kidney called afferent arterioles into each Bowman's capsule and forms tuft of capillaries called glomerulus. The glomerular capillaries rejoin to form efferent arteriole and further forms peritubular capillary network in the cortex around convoluted tubules of nephron. All the capillary network return blood containing useful materials to the renal venules which join to form renal vein that opens into the inferior vena cava. The cortex overlies the bases of medullary pyramids and dips down between them, displacing portions of cortex which invade the medullary territory and are called the renal cortums of Bertini. Cortex contains the straight parts of proximal tubules and the collecting ducts. Renal medulla mainly contains loop of Henle, collecting tubules and ducts of Bellini. The medulla consists of 10 to 15 conical masses called the renal pyramids, whose bases are adjacent to the cortex and whose apices form the papillae. These papillae project into cup-shaped channels called calyces. The urine drains from the collecting ducts of many thousands of nephrons into a calyx. The calyces join to form the renal pelvis, which is the expanded beginning of the ureter, and all of them drain into the pelvis. Ureters. The ureters develop in the region of calyces and connect the kidneys to the bladder. They are muscular ducts which emerge out from the hilum to the kidneys. In adults, the ureters are usually 25 to 30 centimeter long. Urine enters the ureter from the renal pelvis and is conducted along the ureter by peristaltic waves on its walls, propelling the urine from the kidneys to the urinary bladder. Muscles in the ureter walls tighten and relax to force the urine downward away from the kidneys. Urinary bladder. The urinary bladder is a hollow sac-like organ which stores urine temporarily. Bladder has three parts: the apex, fundus or body, and neck. The body has triangular area called trigone. The neck regions possesses two ring-like muscular sphincters called involuntary internal sphincter and voluntary external sphincter, which stops urine from leaking. The ureters and urinary bladder are held in place by ligaments attached to other organs and the pelvic bones. Healthy bladders can store up to 300 ml of urine for several hours. When it is time to urinate, the brain signals the bladder muscles to tighten, squeezing urine out of the bladder. At the same time, the brain signals the sphincter muscles to relax. This helps the urine to exit the bladder through the urethra. This process is known as micturition. Urethra. The urethra is a membranous tube which conducts urine from the lower neck of the urinary bladder to the exterior. The urethral sphincters keep the urethra closed except during voiding of urine. In males, the urethra is about 20 cm long and carries urine as well as spermatic fluid and opens out at the tip of penis by the urinogenital aperture. In females, urethra is about 3 to 5 cm long and carries only urine and opens by the urinary aperture. We shall now discuss the structural and functional unit of kidney, which is known as nephron or uriniferous tubule. Each kidney of an adult human contains about 1 million nephrons or uriniferous tubules, which are each approximately 3 cm long. They filter the blood and produces urine. A nephron consists of two parts: an initial filtering component known as the renal corpuscle and a long tubule called renal tubule, both made of simple cuboidal epithelium. The renal corpuscle or Malpighian corpuscle is composed of an glomerulus and Bowman's capsule. The renal corpuscle filters out large solutes from the blood, delivering water and small solutes to the renal tubule for modification. The nephron begins as a double-walled blind cup called the Bowman's capsule, which is lined by the squamous epithelium. This is surrounded by the glomerular network that receives its blood supply from an afferent arteriole and leaves through the efferent arteriole, carrying the filtered blood. The diameter of afferent arteriole is much more than that of efferent arteriole. The filtrate leaving the Bowman's capsule is very similar to that of blood plasma in composition, as it passes into the proximal convoluted tubule. The renal tubule. It functions as a dialyzing unit. It is attached to each Bowman's capsule and is a long thin tubule with three distinct regions. the proximal convoluted tubule or PCT, loop of Henle, and distal convoluted tubule or DCT. The first region is called the PCT. It is coiled and looped about 14 millimeter long and lined by single layer of cubicle cells and numerous microvilli. It connects to the second region known as loop of Henle, which is a U-shaped or hairpin tube that dips deeply into the medulla. Its primary role is to concentrate the salt like sodium chloride, potassium, etc. in the interstitium, that is, the tissue surrounding the loop. The loop of Henle is differentiated further into descending limb and an ascending limb. The descending limb is permeable to water but completely impermeable to salt. The thick ascending limb of the loop of Henle is impermeable to water but permeable to salt and reaches the glomerulus of the nephron, from which the tubule arises and passes close to its afferent arteriole and efferent arteriole. These limbs operate the countercurrent mechanism to make the urine hypertonic in nature. The third region of the nephron is the DCT, which means that it is farther away from the Bowman's capsule than the other regions. The DCT is similar to the PCT in structure and function. Cells lining the tubule have numerous mitochondria, enabling active transport to take place by the energy supplied by the adenosine triphosphate or ATP. The DCTs from many nephrons connect to a common tube called the collecting duct, which empties into the renal pelvis. It regulates the composition of urine by reabsorbing water, ions and nutrients from the filtrate. This prevents the loss of useful materials and regulates the composition of blood and the body fluids. Though the collecting duct is normally impermeable to water, it becomes permeable in the presence of antidiuretic hormone or ADH. Now, there are two types of nephrons in the kidneys. They are cortical nephrons, which are 70 to 80% in abundance and are close to kidney surface and have a shorter loop of Henle and peritubular network, also called vasa recti. The second type is called juxtaglomerular nephrons. They are 20 to 30% in abundance and are present at the junction of renal cortex and medulla. They have longer loop of Henle and vasa recti. Now, let us discuss the mechanism of urine formation. The renal artery branches repeatedly after entering each kidney, forming smaller and still smaller arterioles. An afferent arteriole delivers blood to the glomerular capillaries for filtration, while efferent arterioles drain filtered blood away from the same glomerulus. The efferent arteriole connects to a second network of capillaries called vasa recti or peritubular capillaries, which are present around the loop of Henle. They reabsorb water, ions, urea and nutrients from the interstitial fluid and help in maintaining high osmotic pressure in the medulla. From vasa recti, blood flows progressively through large veins until it reaches the renal vein, which leaves the kidney and returns blood to the inferior vena cava. The walls of afferent arterioles contain the hormone renin, secreted by juxtaglomerular cells, and macula densa, collectively known as juxtaglomerular apparatus. It acts as a feedback mechanism regulating the renal blood flow and glomerular filtration rate. The juxtaglomerular cells are the site of renin synthesis and secretion. The formation of urine has the following three processes. 1. Ultrafiltration or glomerular filtration of the blood plasma by the glomeruli. 2. Selective reabsorption by the tubules. Here, useful substances such as sugar, salts, water are selectively reabsorbed from the glomerular filtrate to maintain the internal environment. And the third is called tubular secretion, where the tubules secrete certain substances like urea, uric acid, anions, etc. from the blood into the tubular lumen for excretion into the urine. Glomerular filtration of blood. Urine formation begins with filtration of blood through the epithelial walls of the glomerulus and Bowman's capsule. The fluid portion of the blood, which consists of water, urea, ions, nutrients and small proteins, is able to move across the capillary wall. Blood cells and larger proteins, however, cannot cross and are retained in the blood. The molecules that leave the blood, such as salts, water, soluble molecules and urea, enter the glomerular capsule and are called the glomerular filtrate. All of these factors combine to increase the blood pressure and force large quantities of plasma out of the capillaries into the Bowman's capsule and down the tube of nephron. Larger proteins and cells remain in the capillaries, creating a very hypertonic solution. Water and dissolved substances are present in the filtrate at about the same concentration as they are in the blood. Glomerular filtration occurs because the pressure of the blood flowing in the glomerular capillaries is higher than the pressure of the filtrate in Bowman's capsule due to the narrower efferent arteriole. This is known as the glomerular hydrostatic pressure or GHP, which is about 70 to 90 mm mercury. The blood colloidal osmotic pressure or BCOP due to plasma proteins in glomerulus is about 30 mm mercury. The capsular hydrostatic pressure or CHP is caused by the fluid occupying in the Bowman's capsule and is about 20 mm mercury. The capsular hydrostatic pressure CHP along with colloidal osmotic pressure BCOP opposes the glomerular hydrostatic pressure GHP and drives the glomerular filtration. This pressure is known as effective filtration pressure EFP, which is about 25 mm mercury. Hence, EFP is equal to GHP minus BCOP and CHP, which together equals to 75 minus 30 plus 20, which is equal to 25 mm mercury. Both kidneys produce glomerular filtrate at the rate of about 125 ml per minute or 180 liter per day. The selective tubular reabsorption. It is the second process in the formation of urine from filtrate. Much of the filtrate passes out of the nephron tubule and returns to the blood through the peritubular capillaries. As much as 99% of the material in the filtrate is reabsorbed, preventing the loss of useful materials like water, nutrients and ions from the body. As a consequence of tubular reabsorption, urine contains mostly waste materials and excess water. Water and urea are reabsorbed by passive transport, water by osmosis and urea by simple diffusion method. The PCT actively absorbs almost whole of glucose, 75% of amino acids and vitamin C, about 70% of sodium ions, 75% of potassium ions and a large amount of calcium ions from the glomerular filtrate. As the filtrate descends deeper into the hypertonic interstitium of the inner medulla, water flows freely out of the descending limb by osmosis, while sodium chloride diffuses into it until the tonicity of the filtrate and interstitium equilibrate. Longer descending limbs allow more time for water to flow out of the filtrate. So, longer limbs make the filtrate more hypertonic than shorter limbs. Unlike the descending limb, the ascending limb of Henle is impermeable to water, a critical feature of the counter current exchange mechanism employed by the loop. In the ascending limb, the fluxes are reversed, so sodium is actively pumped out of the filtrate, generating the hypertonic interstitium that drives the counter current multiplier exchange along with vasa recta. The vasa recta thus carry the salt and nutrients back from the interstitium to the kidney without interfering with the osmolarity gradient. This hypotonic filtrate passes to the DCT in the renal cortex. The sodium and chloride ions are actively pumped from the ascending limb of Henle while water is retained. Since the ascending limb is impermeable to water, this creates a concentration gradient in the medulla, in which the concentration of sodium and chloride ion is greatest in the region of the bend of loop. The ascending limb of Henle actively reabsorbs about 25% of potassium ion and some chloride ions. DCT and the collecting tubule actively reabsorb some sodium ion from the nephric filtrate in exchange with potassium ion of the interstitial fluid. The counter current multiplier system is an active process which is responsible for the production of concentrated urine and preventing loss of sodium and chloride ions from medulla in the collecting ducts of the nephrons. The exchange occurs between Henle's loop and vasa rectae. Fluid passing from the loop of Henle to the distal tubule is less concentrated than that entering the loop, but because of high osmotic pressure in the medulla, water diffuses out of the collecting ducts, producing concentrated urine. This helps in conservation of water. As the nephric filtrate moves in the nephron towards the collecting tubule, about 99% of water, whole of glucose and amino acids, most of sodium and chloride ions, some urea and uric acid are reabsorbed back into the blood. This process, by which only useful substances are reabsorbed from the nephric filtrate, is called selective reabsorption. Tubular secretion. It is a process by which the glandular cells of the nephron, especially the DCT, extract the waste like uric acid, creatinine, hippuric acid, potassium ion, proton ion, ammonia from the blood in the capillaries by active transport and secrete them in the nephric filtrate. Most of the potassium ion eliminated in urine is secreted by DCT and collecting tubules in exchange with reabsorbed sodium ion. The secretion of hydrogen ion is an important way in which kidneys help to control the pH of blood. Besides food preservatives, medicines, harmful drugs and metabolic byproducts such as creatine are removed from the blood by tubular secretion. As a result of these three processes, the wastes are changed into urine. Regulation of urine formation. The urine formation and excretion is under the control of nervous system and endocrine glands. The nervous system. The sympathetic nerves control the glomerular filtration and tubular reabsorption. Hormonal control. Aldosterone produced by the adrenal cortex helps in reabsorption of sodium ions and excretion of potassium ions in the tubule. Antidiuretic hormone or ADH or vasopressin secreted by the neurohypophysis. Its active secretion increases reabsorption of water resulting in concentrated urine. Parathormone secreted by parathyroid gland enhances absorption of calcium and increases glomerular filtration rate. Functions of excretory system. The kidney's primary function is the elimination of waste products from the bloodstream by production of urine. They perform several homeostatic functions such as maintaining the volume of blood, interstitial fluid and pressure of extracellular fluid, maintaining the ionic balance and osmotic concentration in extracellular fluid, excreting the toxic metabolic byproducts such as urea, ammonia and uric acid, helping in water conservation by preventing water loss, removing the nitrogenous waste from the body, maintaining the acid-base homeostasis by regulating the pH of the blood. The function of two kidneys can be carried out by one healthy kidney if needed. Some people are born with only one kidney. Other people choose to donate a kidney. A kidney may be severely damaged by disease or injuries which requires removal. The accessory excretory organs. Liver. The liver detoxifies and breaks down chemicals, poisons and other toxins that enter the body, like transforming ammonia into urea and uric acid. The liver also produces bile, which helps to break down ethanol, fats and other acidic wastes including ammonia into harmless substances. Large intestine. The large intestine collect usable water and solid waste from throughout the body, and the waste is excreted out from the rectum in the form of feces. Skin. In mammals, the skin excretes sweat or perspiration through sweat glands throughout the body. The sweat eliminates water and salt, evaporates and helps to keep the body cool when it is warm. The sebaceous gland secretes sebum containing sterols, waxes, fatty acid, etc. and helps to keep the skin moist and soft. Lungs. It helps in the elimination of carbon dioxide and water vapor during expiration process. The metabolic waste products are removed from the body by the process of excretion. The nitrogenous wastes, inorganic salts, excess vitamins, pigments, etc. are removed by the kidneys, while carbon dioxide and water vapor are removed by the lungs. Different types of nitrogenous wastes are found in animals depending on their habitat. In humans, the waste is urea, which is produced in the liver by a process called ornithine cycle. The excretory system, comprising of kidneys, ureters, urinary bladder and urethra, form the urine by filtering the blood. The functional unit of kidney is known as nephron, and this structure helps in the urine formation by 3 stages, that is glomerular filtration, tubular reabsorption and tubular secretion. The high threshold substances are reabsorbed completely from the glomerular filtrate, while low threshold substances are reabsorbed in much smaller amounts. The function of the kidney is regulated by the nervous system and the endocrine glands. The kidneys play an important role in maintaining the water and osmotic concentration of blood. Skin, liver, lungs and intestine are other important excretory organs in man.